My name is Randall Platt, and I'm an associate professor at the ETH Zurich in Basel, Switzerland. And my research is focused on developing gene editing technologies for both diagnostics and therapeutics in the area of intestinal health. And so to do this, we use the language of genetics, molecular biology, synthetic biology, microbiology, computational biology. So on November 9th, 1989, when the news came out of the fall of the Berlin Wall, I was very likely in the arms of my mother in Salt Lake City, Utah. And while I was clearly too young to remember this day, the significance of the event permeates my everyday life and biomedical research. Whereas before the fall of the wall, science was compartmentalized, but afterwards, barriers began to dissolve, and scientists were not only encouraged to work together, they started to thrive and solve some of the world's biggest challenges. And this is what the fall of the wall means to me. The gut and its microbiome are central to health. This organ converts food to fuel for your body and is interconnected with numerous body systems. Dysfunction of the gut can lead to problems in metabolism, in the immune system, and even the brain. And the importance of the gut has been well known for a long time. But the mechanistic basis of how changes in what we eat or what we experience and how these lead to disease are poorly understood due to the lack of ability to make direct measurements inside the body. And so to overcome these challenges or break down these walls, we've developed engineered bacteria with memory. And the point is that these safe cells have the ability to remember what they experience. And so if we take these safe cells and we feed them to one-day people, the idea is that they'll traverse the gastrointestinal tract. They'll interface with your body, sense health, sense disease, record this information. And when they leave the body, we can sequence these cells and make sense of what was happening. We can understand how food is connected to health. We can understand which people are sick and which people are not. And so we are using these engineered cells to break down the lack of ability to make measurements inside the body. Now that we have these engineered bacterial cells with memory, my goal is to operationalize the technology in people to help diagnose disease and guide health. Like the microscope enabled the field of pathology, we believe that engineered bacteria have the potential to create a new way of measuring health and guiding the development and implementation of new medicines. However, the biggest walls that stand in our way have to do with both the regulatory approval path and commercialization of a fundamentally new type of technology. Nonetheless, we are dedicated to breaking down these walls and fully realizing the potential of these engineered cells with memory for the improvement of health and well-being of people around the world.